There we go.
Great, you're good.
And do you see any live transcript? Or here we go, request, here we go. I am requesting live transcript. Okay, and I think the waiting room's already in the session. I believe
So do I have to enable?
You should be able to enable live transcript also next to record. So
Okay, there we go.
We're good.
Great. So it is 2 o'clock and I think we are ready to get going.
Yes.
Michelle, please.
Oh, good afternoon. Today's symposium presentations are, as the session is titled, Newly Developed Models for Amylogenesis. I am Dr. Michelle Cortez Salva. I am the health program specialist at the Integrative Biology and Infectious Diseases branch at NIDCR. Jason?
Hello, good afternoon or good morning, everyone. My name is Jason Wan. I'm the program director at the National Institute of Dental and Craniofacial Research, overseeing the Mineralized Tissue Physiology program. So first, just some rules for this session. This session is recorded and please make sure you mute yourselves if you're not talking. The chat will be monitored by Dr. Michelle Cortez. She'll be looking at the chat, and if you have anything, comments for the discussion session, you can also use your raise the hand emoji. First of all, I'd like to thank the Mineralized Tissue Group for sponsoring this symposium. And I guess just to launch into this particular session, I'm going to give just a brief background about how this all started for these amylogenesis models. About 2.5 years ago, NIDCR issued a opportunity and initiative with the purpose of developing these novel amylogenesis models. And so the RFA was a two-phase activity which incorporated 2 objectives. So one was to have the generation of new and improved models to closely mimic physiological enamel development and maturation in order to enable the studies of amylogenesis, followed by a second phase, which was to validate those models. So this is a two-phase, two-prong objective via a cooperative agreement with our vision that these models and data that would be generated would help understand enamel malformation and diseases. And so we can begin to reproduce and repair enamel. And part of the reason was to have this data available for the community to use. So Michelle, turning it back to you. Michelle, you're on mute. Still cannot hear.
Hello, yes.
Yeah, there we go.
For today's session, we have 4 speakers: Dr. Mirali Pandya from Texas A&M University, Dr. Jan Hu from the University of Michigan, Dr. Ophir Klein from the University of California, San Francisco, and Dr. Michael Payne from the University of Southern California. The session will be, the presenters will present for 50 minutes, and after the presentations, there will be a Q&A/panel discussion session where Dr. Rob Schuller from the University of Southern California will provide with a short description of FaceBase, which is the data repository where the PIs are depositing their data. We also encourage the participants and attendees to put your questions in the chat or raise your hand and unmute yourself to ask the questions during the Q&A discussions of the session. And now we can present our first speaker, Dr. Mirali Pandya. Go ahead, Mirali, share the screen.
Okay, you can all view my screen. Yep.
All
Yep.
right, thank you. All right, good afternoon, everyone. I'm Dr. Mirali Pandya, and I've had the pleasure to work on enamel for the last 6 years with my mentor and my guide, Dr. Tom Dequish, here at Texas A&M. And we worked on understanding the enamel formation as a focus, which was also my focus during the PhD and after. And today I'm excited to share with you 2 of the novel model systems for a deeper understanding of amelogenesis. So these are the 2 models that we had proposed for the RFA. The first one that was proposed was to understand the transportation of mineral ions through ameloblasts. And for that, we are using a Clathrin mouse model. And the second model that we proposed was to use this unique 3D bioreactor to basically explore and develop a new way to culture these ameloblasts in vitro. So let's take a look at the first model, which is the Clathrin-mediated ion transportation during amelogenesis. And so I think this is probably a very common question that a lot of enamel researchers have in their mind. Probably at some point or another, a lot of them have also tried to work on it. And the question is that how do these mineral ions from the blood vessels that we have in the stellate reticulum get transported all the way through these ameloblasts and get deposited in this enamel layer? This really short distance is still like a black box, and the exact phenomenon as to what's occurring in there is still not clear to us. So here are a few of the models that have been explored so far. The first one is the calcium channel model that we have, which basically proposes that these ion channels are responsible for the transportation of these ions through the ameloblasts. There's another model, which is the interstitial transport model. And that is the model where the ions are basically transported between these 2 ameloblast cell layers and via cell movement. And then we have a third model, which is a vesicular transport model, where the secretory vesicles in the ameloblasts might be transporting these mineral ions. So to begin our quest, we first looked at an amelogenin null mouse model just to see what's happening, what's going on there. And a co-immunoprecipitation assay demonstrated that there is an interaction between the amelogenin and Clathrin at some point. So we did get a protein-protein interaction, and there was a reduction in the protein, Clathrin protein, basically in these amelogenin knockout models when compared to a wild type. When we looked at the histology from these mice, what we saw in the ameloblasts is that it stained very strongly for a Clathrin antibody at these, specifically in the secretory vesicles within the ameloblasts. And if you look at the null model, which is to the right, which is the panel D, there we can see that the immunoreaction wasn't as strong for the Clathrin, which basically corroborates what we found with our Western blot, that there was a reduction in the Clathrin protein. Right, so I think the question here is, what is a Clathrin vesicle that we are talking about? So in a cell, a Clathrin is in a triskelion form, where it's in the cytoplasm, and it basically binds to an adapter protein that has bound membrane, linking one of its 3 feet to these membrane one at a time. So Clathrin cannot bind to a membrane or a cargo directly, but it will use these adapter proteins to do so. And this triskelion will then bind with another membrane attached triskelion, and then they will form a lattice of these hexagons and pentagons that is probably like, it looks like a soccer ball, right? I mean, that's what it is reminiscent of. And that is what helps to pull the membrane into a bud. And so at this point, we hypothesize that the enamel proteins and minerals are transported throughout the ameloblasts in these triskelion clathrin-coated cages. So to address that question about what is the extent of the clathrin involvement during amelogenesis, we proposed a mouse model, which was a clathrin conditional knockout model to test our hypothesis. We crossed these conditional knockout models with two different amelogenin Cree models that we had available. One was a Cree from Dr. Kulkarni's lab, and the second Cree was from Dr. Papageraikis' lab. And after eagerly awaiting for some results and after multiple breedings that we had, we were finally able to successfully generate a conditional knockout model with the amelogenin Cree models that we crossed. The semikin sections of these knockouts demonstrated these. I'm not sure if you are able to see my mouse where it is, but if you look at figure
we can
A,
see it.
Oh, okay, perfect. Thank you. So if we look at figure A and figure B, so we are talking about the secretory vesicles that we see, and here is these really voluminous secretory vesicles that we saw in the knockout models. And this wasn't just with one Cree that we crossed. So this was Dr. Kulkarni's Cree, and this is Dr. Papageraikis' Cree. And we saw the same results with both Crees, very identical to each other. And getting a closer look or a higher magnification look with an SEM, we were able to see the enamel prism structure. And what was different in these clathrin knockouts that we noticed is the lack of these prism structures right around the dento-enamel junction area, which we can see with both Crees. They look a little different because they were done with two separate SEMs at two different labs. Okay. Okay, we understood that there was a change in the prism structure, but what exactly had changed in them? We do know that it's made up of calcium and phosphorus, so one of these materials had to change, right? So we did an elemental analysis which demonstrated the amount of calcium and phosphorus in the knockout group, and we noticed that the knockout group had less phosphate, specifically around that DEJ area, where it wasn't, which is visible right here in the figure H. And the green basically shows the calcium amount that is present, and the red is the phosphorus. And here we can see it's probably a complete band of green with a significant lack of the red that we see here in a wild type model. So in order to summarize what we have, we basically crossed our clathrin conditional knockout model with two different Cree models from two different labs. And we got a very similar phenotype with both of these Cree models for our clathrin mutant flux mice. The good news is that this mouse has now been deposited at the MMRRC, so it will be available for the enamel community to use and to start their own research if they find it interesting to use this Cree. We found these really large enlarged secretory ameloblast vesicles in the mutant mice. And there was a lack of prism structure, which was observed at the DEJ, and there was a reduction in the phosphate in that enamel layer, specifically at the DEJ. So I'm switching gears now a little bit, moving on from a mouse model to an in vitro 3D bioculture model. So for this, we used a bioreactor. Now, a bioreactor is basically a rotatory cell culture system where we have a horizontally rotating cylindrical tissue culture device. This was initially developed by NASA to grow cells in space and to do their experiments, but luckily it's now available to all the scientific community to use. And this device enables us to basically grow the cells in a three-dimensional structure. So one would ask, okay, what is the point of using a 3D bioreactor model compared to what we already have? So here are some of the advantages that I listed. So one is the ability to co-culture multiple types of cells. So you're not limited to just a feeder layer and then another layer on top. If you want 3 different cell types, you can still do that with this. Another thing that we have is that it can be used with or without a scaffold to generate 3D cell models. So it can also be used for tissue culture as well as cell culture. It has a very low shear force in the rotating vessel, which allows for these spheroid growths. And there is a high mass transfer of nutrients in the media, which is one of the problems that we face with the 2D culture. And this helps prevent cell death within that spheroid core and enables us to culture these cells for a very long time. Here is a closer look at the bioreactor. So as we can see, we have 4 different vessels that we can conduct 4 different experiments at the same time within these vessels. They each act as a separate unit. That's what we did. We ran our different models at the same time so that we could maintain similarity between experiments and just make sure that the environment and everything that we are providing is at the same time. Here is a closer look at the vessel. So this is how it looks like. This is where we connect our syringe just to ensure or change our media, and then they are closed just to, and this is a valve basically, which stops anything from going in or out. And this is just a scaffold that we initially used or that came with the bioreactor. So this was a PLGA scaffold that we had, and this is the one that they suggested that we use with this bioreactor. We did not end up just using that because it did not work very well for our experiments. So apart from that, we also tested, I would say, about 7 or 8 different scaffolds before we found a few that we liked. And then there was one that we honed into, which was this porous collagen scaffold. As you can see, it's a very porous structure. And this was the one that gave us really good results for the experiment. And it was a collagen-based scaffold that we have. So we place these scaffolds right inside this bioreactor. These are the syringe ports. Here is an oxygenator membrane right there, which constantly ensures that there is an exchange of matched nutrients, and it helps us enable cell growth for a very long time. At no point is this touching this membrane, because this is always in constant motion and it's rotating. So we based our model off of Dr. Ophir Klein's JOVE paper, where he harvested these cervical loop cells, and that's what we did. We modified it just a little bit, and those are the primary cells that we used to co-culture our system. So here is just a closer look at how it looks within a mouse mandible. Here is the co-culture of the cells and the matrix. And what we found was that if we tried to just culture the cervical loop cells, we did not get a very good expression of amylogenin, but we did see that with a co-culture. And here is just a cross section of how this spheroid looks like and the cells within expressing amylogenin. So this is an immunoreaction for amylogenin. So the first result that we got was these elongated tube-like cells within the system. And that's how they looked when we cut them with our paraffin knives. And this was not what we were aiming to have. Along with these elongation, we also need the cells to be polarized and to look like the actual ameloblast that we work with. So we changed our approach a little bit, and we tried to focus more on the scaffolds that we were providing for these cells, because that's actually their home. That's where they live, that's where they breathe, that's how they interact with each other. And that's what we focused on. So we tried 3 different matrix gels combination for this scaffold. So the scaffold that we used was still the sponge gall scaffold, and then we coated them with these different combined groups. And here, as you can see the results that we got. So this is a live dead cell imaging that I did. So this is for the LRAP group. This is for where I just used the matrix. And here is what I received with the combination group. And here is a histology for the cells that we had with these 3 different groups. And as you can see, the combined groups, they give us these really nice polarized, elongated amylogenin, amyloblastin producing cells. A closer look at these elongated and polarized amyloblast-like cells. Here are these really long processes that we have. And that's what we saw after we cultured our cells with the combined matrix and all of the media components that it needed to make this amylogenin and amyloblastin. So what we found was formation of elongated and polarized cells. We had an enlarged cytoplasm, and they were reactive for amylogenin antibody. And as you can see, here is a staining for amylogenin. This is just a control for my immunoreaction. And we not only the cells expressed amylogenin, amyloblastin, we were also able to see expression for MMP20 and KLK4, which we know is very important for this. So we got 2 different cell populations. These long elongated cells were the ones that did express these amylogenin related proteins. And these tiny little cell population that we had, these were the ones that were negative for any kind of amylogenin proteins or any stainings. To further check, the current resources that we already have available for the enamel community, we had these head cells that we also tried to see how they did with our bioreactor system. And here are these head cells that we did try and grow in the 3D environment. They were positive, they did produce amylogenin, but what we saw was a lack of these polarization and elongation, like we saw with our cervical loop and dental pulp co-culture combination. So if I went through it very quickly, and if you would like to see a detail of exactly what happened throughout these, or what was the exact media that I used and every protein that I used, we were able to publish these papers and the protocols. And here are the publications that are now available online. And we were very lucky to get a cover page for the Stem Cells and Development Journal to basically focus on some of our beautiful ameloblasts in there. So just to summarize, all right, sorry,
Okay.
I promise I'm at the very end.
Okay.
Just to summarize, or just focus on what is the advantage of this. The cell culture models reduce the variability that is observed among animal strains, and it also enhances the reproducibility that we have among different labs. So we can share this with anybody who would want to start these experiments. We have already published everything that we had, and we are happy to answer any more questions that anyone else might have with this. So as far as our current work goes, we are collaborating with Dr. Klein's team to get a few CRE lines, and we will also conduct some long-term bioreactor studies to characterize our model, and we will also determine the modes of ameloblast ion transport further using our Clathrin model. And I can't end this talk without saying thank you, Dr. Won and Dr. Cortez, for your tremendous support that we have received throughout our UG3 phase and that we are continuing to receive for the UH3 phase. It is greatly appreciated. And here's our team. The work is still undergoing and everything's still under construction.
Thank you very much, Dr. Pandya.
Bye.
Our next speaker will be Dr. Jan Hu. Can you share your screen?
Yes, I'm working on it.
Yeah, okay. Go ahead, Dr.
Okay, good afternoon, everyone. The Michigan project is to develop and validate novel immunogenesis models. On behalf of our co-authors, I'd like to declare that we have no conflict of interest. This project is funded only by the NIDCR. The objectives of this study, short-term, is to develop well-characterized mouse models with human mutations causing immunogenesis imperfecta, or AI. Long-term, we hope this study findings would allow to advance our understanding of enamel formation and biomineralization, and to help provide the basis for genetic testing and to facilitate interventions of patients with AI. Enamel forms in stages under precise genetics and molecular controls. In this study, we target two specific genes, SOC13A5 and ODEF. SOC13A5 has an early onset of expression and sustained throughout immunogenesis, while ODEF expression has a very narrow window limited to transition early maturation stage. These molecules, when mutated, will result in human AI. Therefore, they are important for enamel formation. However, their precise function in facilitating enamel formation is largely unknown. This study has two specific aims. First, to establish and validate SOC13A5 knockout mouse model that's homologous to human AI. And then second, to generate SOC13A5 flat tag knock-in mouse model so that we can precisely localize the expression of this particular molecule. There are no commercially available valid antibodies against SOC13A5 gene product. The second aim is to develop and validate ODEF knockout mouse model homologous to human AI. To validate and characterize the proposed mouse models, we will use in situ hybridization to define expression of these genetic gene molecules. We use dissecting microscopy to establish analysis of the gross anatomy. We use histology, immunohistochemistry to define protein expressions and also assess cellular and tissue integrity. To characterize the structure of enamel, we define its surface roughness, susceptibility to attrition, thickness, rot pattern, hardness, and ultrastructural features using backscatter SEM, nanoindenture, and focus ion beam SEM. M1 is to generate two models associated with SOC13A5. This particular gene encodes a sodium-dependent citrate transporter. Citrate is very important facilitator for energy homeostasis. It is also critical to facilitate the transformation of amorphous calcium phosphate into hydroxyapatite. In the body, there's a significant amount of citrate associated with bone and in circulating blood. In the absence of functioning citrate transporters in human, it will result in developmental and epileptic encephalopathy 25 and immunogenesis imperfecta, where affected dentition has almost no enamel, therefore severe hypoplastic AI. Citrate in the developing enamel has been carefully defined by Dr. Yasuo Yamakoshi using colorimetric assay. In the taking the porcine developing enamel, he determined that there's an average of 22 micromole of citrate in there. As the enamel continued to mature, the citrate concentration increased. Using in situ hybridization demonstrated that SOC13A5 is expressed by aminoblast from pre-secretory to secretory and to maturation stage. It is also expressed by odontoblast and osteoblast in the developing alveolar bone. We use CRISPR-Cas9 gene targeting strategy, introduced a single nucleotide change that altered codon 337, arginine to premature stop codon. In that process, we also introduced the 6 nucleotide change that did not change codon in order to facilitate genotyping. Following the gene targeting, we screen for chimeric mice with the positive alleles. Those positive chimeric mice were bred with B6 wild-type mice. After 2 generations, then we subjected positive mice with correct alleles for Sanger sequencing. Sanger sequencing of heterozygous and homozygous SOC13A5 knockout established that the correct genetic targeting precisely established in this particular mouse model. Under the dissecting microscope, we can appreciate hypoplastic AI in the null mice, while there's no morphological differences between the heterozygous and wild type. The teeth are soft, as you can see. Following a few weeks of occlusion, there's a significant attrition. Histologically, the defect can be appreciated starting early secretory stage, where enamel matrix deposition, as you can see in the heterozygous and wild type, as this darker red line, is not evident in the null. And these defects continue to worsen until progressing into the maturation phase. There's no mineralized enamel, but only the ectopic calcification nodules. In situ hybridization established that the wild type as well as the mutant transcripts were expressed in these mice. To assess the enamel structure after maturation, we look at cross-section of mandibular incisor and compare many mouse models with hypoplastic AI phenotype. The enamel features of SOC13A5 knockout is very similar to those defects that have been observed in the enameling absent, aminoblasting absent, and the acid phosphatase protein absent mouse models. In order to assess cellular integrity and enamel ribbon formation, we use FIPSAM to analyze the cell and mineral formation. This is the odontoblast layer, pre-dentin, mineralizing dentin gradually increase in thickness, and this is a layer of amyloblast cells. Typically, around this stage, enamel matrix would have been deposited, and enamel ribbon formation can be appreciated around here. So we're going to look at higher magnification of this region in the knockout as well as wild type. So at this particular developmental stage in the wild type, we can appreciate elongating enamel ribbons that have been organized into rod and interrod structures. In the SOC13A5 knockout, these are 2 consecutive areas. Initially, we can see sporadic enamel ribbon formation, and then, however, very rapidly after their detection, they are completely disappeared. leaving the interface of the mineralized dentin and amyloblast cell membrane empty without enamel matrix or enamel ribbons. So in summary of the first model, the accurate gene targeting has been accomplished and validated. We completed morphological assessment at the of the gross anatomy and histological level. We are now continue to assess ultrastructural features of this mouse model using the proposed experiments. The second model in M1 is to generate a flag-tagged SLC13A5 mouse model. And we use conventional homology arm recombination strategy to introduce nucleotides in code 8 amino acid of flag tag to immediately upstream to the natural stop codon. This is extremely difficult procedure. However, we were able to, after 3 tries, able to introduce successfully the flag tag right before the THE stop codon. We have to analyze how the introduction of flag tag may have impacted growth and development. In terms of tooth development at the gross anatomy level, there's no appreciable differences in terms of tooth size, morphology, color, and shape. At the histology level, we analyze the secretory and maturation stage. The development of these 3 genotypes seems to be comparable. In situ hybridization established that the transcript expression of a wild type and the flag containing transcripts were present at comparable level. Using commercially available antibody against flag, we were able to detect the expression of the C-trait transporter with a flag tag. This is showing the secretory stage amyloblast, positive standing throughout the cells, but perhaps most prominently along the distal membrane surface. At the higher magnification, this is the wild type for comparison purpose. We can see significantly higher expression on the distal membrane of amyloblast cells at higher magnification demonstrated the positive signal is associated with the distal end of Tome's process, where secretion and endocytosis activities are extremely prominent, therefore require energy consumption. Summary, we have accurately targeted this particular model, and morphological assessment at this time did not show any gross level enamel or tooth defect. We are continuing structural analysis at ultrastructural level. M2 is to develop the ODEF knockout, mimicking human AI with ODEF mutation. A decade ago, it was established that ODEF absence would result in hypomaturation AI, established the importance of this protein for immunogenesis. In human, the gene encodes ODEF localized to chromosome 4q21 region. It is in between the 2 groups of secretory calcium-binding phosphoprotein important for enamel, dentin, and bone formation. We use CRISPR-Cas9 technology to target a single nucleotide change of codon 41, changing cysteine to premature stop codon. At the same time, we also altered 6 nucleotides to allow genotyping. After 2 generations of breeding of positive chimeric mice, we were able to establish a Sanger sequencing, confirming complete accurate targeting. In situ hybridization established that ODEF expression is low, and it is only detectable during a very narrow window of time, that is post-secretory transition to early maturation phase of aminogenesis. In the absence of ODAF, hypomaturation AI would occur. Enamel thickness is of normal range, but enamel can fracture in chunk after occlusion. Histologically, secretory stage, there's no appreciable differences, but as development progress, we can see flattening of amyloblast cells and enlarging of enamel surface space and development of a cystic-like change. These changes can also be appreciated on the lateral section of mandibular incisor in the wild type right at the interface of post-secretory transition and early maturation phase. Amyloblast cells are detached from the enamel matrix surface. They became shortened, flattened, and there's a cyst formation. How would those changes impact mineralization of enamel? We assess the degree of mineralization using backscatter SEM at level 8 right before incisor eruption. We can appreciate that among these 3 genotypes, enamel thickness is not disturbed in the ODAF knockout. The rod and interrod structure is very nicely laid out. However, the degree of mineralization is completely disturbed. Compared to 2 other hypomaturation AI model, this is perhaps the most severely affected hypomaturation AI in ODAF knockout. It is corroborated.
Sorry, can you wrap up?
Okay, it's corroborated by hardness testing data set. These cellular changes leading to apoptotic cell death of amyloblast can be appreciated in FIP-SAM data sets. We have successfully targeted this mouse and have completed initial assessment. At this time, we believe that SOC13A5 flag is extreme. This model allowed us to precisely localize citrate transporter to the distal end of amyloblast, establishing its importance for aminogenesis. ODAF expression is required for amyloblast to transition from secretory into maturation stage. These data sets of those models are available at FaceBase, and 3 models plus additional mutation models have been deposited successfully at MMRC. I would like to acknowledge our research lab collaborator as well as funding from NIDCR. Thank you.
Thank you, Dr. Hu, for your presentation. Next speaker is Dr. Ophir Klein.
I'm
Dr.
sorry.
Klein?
Stop sharing.
Okay.
Okay. So you see the presenter view or the right screen?
Presenter
You see the
view.
full presentation.
You see the full, you see the correct screen. Okay, great. Well, yeah, thanks to Jason and Michelle for the opportunity to speak to you. I'm going to tell you about a project that we've been doing, which is highly collaborative. And I'm just going to tell you about part of it, which is developing this suite of mouse reagents to study amelogenesis. Another part of what we're calling the Enamel Atlas is a biophysical characterization, which is in collaboration with Dirk Yoster's lab, and I won't talk about that today for the sake of time. And I just want to mention the folks who have done all the work, who are particularly Adya Verma and Tom Wall, together with Oscar Casares, who are members of my lab. And this is a collaboration with Jeff Bush at UCSF and with Dirk, as I mentioned. So as you're all familiar, just moving you guys out of the way. And for the formation of enamel is a very complex process, and ameloblasts undergo several stages of differentiation, which are defined by morphological features. And the molecular signatures and programs underlying these are still poorly understood. And that's one of the long-term goals that we have, is to understand these. And so the cells go from progenitors. This is using the mouse incisor ever-growing model, but the same steps occur in rooted teeth as well. The progenitors will make the secretory, the pre-secretory and then the secretory ameloblasts, and these then mature, which is when the proteins are removed and there's secretion of a large amount of ions and hardening of the calcium phosphate. And so one of the goals that we have is to develop tools that will enable us to study these various stages of ameloblast maturation. There are a few reagents that are currently available that many labs, including ours, have taken advantage of. And these include the keratin 14 and keratin 5 Cres, as well as inducible Cres or Cre-ERs. There are some challenges with these, including that they are very widely expressed, so you can't get any stage-specific resolution related to these various phases that I mentioned before. And also because they're widely expressed, if you're knocking out a gene, there can be high levels of lethality because it can affect other organs as well. There is one ameloblast stage-specific Cre, which is the amelogenin Cre, but this is a transgene as opposed to a knock-in, and it's constitutive, it's not inducible. And there are currently, to our knowledge, no available conditional alleles. And so because these enamel matrix proteins are the basis of the extracellular matrix, we then set out to make various reagents to study these. These are secreted during the secretory stage. They're processed by MMP20, which is an enamel-specific protease. And then during the maturation phase, there are additional proteins that are secreted to kind of finish off the process. And then there's a degradation of the matrix that occurs by KLK4. And the critical importance of these enamel matrix proteins has been documented through making complete knockouts. These is documented here, and I apologize for leaving out the references just for the sake of space. The mutations often result in severely impacted and completely collapsed enamel, which instead of forming the typical 100 micron thick structure, just remains a very thin layer of disorganized crystallites. Or in the case of the KLK4 knockout, it's severely hypomineralized, which again affects our ability to understand the underlying molecular pathways that are responsible for amelogenesis and how the proteins work together to form enamel. So what we've been doing is generating a series of novel mouse models, which we are making. Many of these are already available to the community, and the remaining ones will soon be available in order to be able to study amelogenesis from various perspectives. So we're making conditional or flox alleles of the major enamel matrix proteins and proteases, which will enable studying these proteins' role during amelogenesis at a high resolution and in fact at a single cell level. Stage-specific Cre drivers as well as fluorescent reporters, and then mutants of the self-assembly domains of amelogenin that I'll talk about in a minute. And the process that we use for this is CRISPR-Cas9 mediated recombination. This has actually been very efficient for us. We've made all of these lines in-house, and they actually have gone fairly quickly considering the large number of alleles that we've generated. So in terms of the conditional alleles, we chose 6 genes, the 6 shown here, to make conditional flox alleles. These can then be combined with existing Cre reagents as well as the new Cre reagents that we're generating. If they're combined with inducible Cres, then you can get mosaic Cre activation, which will enable many groups who are interested to study the question of how these proteins function within individual ameloblasts or clusters of ameloblasts. And making these was not trivial because they have this shared evolutionary history where they're located on one chromosome and they underwent neo-functionalization after duplications. So what that means is they have similar genomic structure, and it wasn't possible to just target sort of random exons without thinking this through. Because the first coding exon contains the 5' UTR, the secretion signal, and the amino acids for the mature protein, we usually targeted either the first methionine or the secretion signal. And these have turned out quite successfully. So here I'm showing you just the sort of test case where we crossed the conditional alleles with a beta-actin Cre, which recombines ubiquitously, to show you that we were actually able to successfully recombine these flox alleles. So these four genes that I'm showing you here now all have conditional alleles. That the unrecombined alleles have wild type enamel. They don't have any phenotypes, which suggests that there's no disruption of the endogenous function. And now we're analyzing the effects by recombination. But essentially, these alleles work. For the proteases, we targeted either exon 2 to generate a frame shift in exon 3 and a premature stop codon, or that was for MMP20, or for KLK4, we targeted the first methionine to prevent expression. And again, here we crossed them with the ubiquitous Cre driver, and we see that the recombination worked. So those were the conditional alleles. Now moving on to the Cre drivers and the reporters, there were two ways that we could do this. One was knock-in, knock-out, or knockout, knock-in approach, in which the protein of interest replaces the endogenous gene, and so you don't have any expression of the endogenous gene. And the other way was to use a P2A in which the endogenous gene is expressed, and these are expressed in as an equimolar ratio of proteins with the construct that's the gene of interest and the endogenous gene. And so for various reasons, we chose one of these approaches. Here I'm showing you the amyloblastin and amelogenin Cres. For the amyloblastin Cre, we used a knock-in, and whereas for the amelogenin Cre, we used P2A because there's only one copy in males since it's on the X chromosome, so that enables the preservation of endogenous function. And you can see both with the reporter assay histologically as well as with flow cytometry that this was very efficiently recombining, and we could sort out a large number of cells, which will be useful for the projects that I'll tell you about in a minute. The amyloblastin Cre ERT2, this is an inducible Cre, also works quite well. It marks specifically the secretory stage amyloblast following a single tamoxifen injection. The efficiency is lower, as would be expected, than the constitutive Cre, but again, this will enable the analysis of secretory stage specific function of genes, where you could target essentially individual ameloblasts. For KLK4, we made both the constitutive and the inducible Cre to label the maturation stage ameloblast. Again, the Cre itself is highly efficient, and you can see that both from the sections as well as from the sorting. The ERT2, again, as we would expect, is a little bit less efficient. This is following just a single dose of tamoxifen injection. We can make this more efficient if we inject more tamoxifen, but we do feel like there are potential benefits to this mosaicism as well. Now moving on to the reporters, the amyloblastin GFP marks specifically the secretory stage ameloblast. This, I just want to show you this experiment that we did here, which is actually a really beautiful result that Tom and Adia and Oscar got, in which they crossed the amyloblastin GFP with the KLK4 Cre tomato reporter. And you can see this really beautiful transition visually from one stage to another. And this can also be seen when we sort these cells out. So I think this will be really useful for people who want to isolate specifically from the same mouse different stages of at the ameloblast life cycle. And then for the last couple of minutes, I'll just tell you about a more functional project that we've done. So this is also a tool which we've generated, but one from which we've also obtained some initial results. And it's based on earlier work that we and collaborators did looking at ameloblastin, which forms these higher ordered structures via a unique self-assembly motif, which is not present actually in any hard tissue except for enamel. And it consists of these 3 hydrophobic amino acids within a stretch of 6 amino acids. And when these were mutated in the mouse, and this was now published a few years ago, the enamel was still formed, but you can see comparing the mutant to the wild type that the prismatic and interprismatic enamel fuses and the individual crystallites are not well defined. And when we looked at the amelogenin protein, we found that there were 2 copies of similar stretches of amino acids similar to the ones that we found in ameloblastin. And so we wanted to investigate the role of these clusters of amino acids and specifically how they affected the smaller proteins that are liberated from the larger amelogenin protein. And so we made mutants in these two clusters. And preliminarily, what we found, so the targeting was successful, and they make normal amounts based on sorting of the EpCAM positive cells in qPCR of amelogenin. And then when we did micro-CT analysis, what we saw was that there was severely hypomineralized incisor in the E3 mutant, whereas the E5 mutant looked fairly similar to control. So the E3 mutant actually looked almost like the amelogenin mutant, the constitutive mutant. And then when we looked with higher resolution micro-CT, we saw that there was largely missing enamel and the formation of these crystallites that were present here. Whereas in the E5 mutant, the structure is mainly preserved, although we're still doing some higher resolution analysis of this. And so just to wrap up, those are the reagents that we've generated. We still have another couple that are in the queue, and they should be ready hopefully very soon. And as I mentioned, all of these will that are produced are available to the community. They're being deposited in repositories, but we're also very happy, since it's a relatively small community. If anybody wants them, we've already worked on sending them out to a number of labs, and please just drop me an email. But they are going to be available through these repositories, and we're cataloging everything on FaceBase so people can see this. We're now moving on to use these reagents to try to understand some more about amelogenesis. One of the first things we're doing is to continue building this enamel atlas. We're performing transcriptional profiling of the later stage ameloblasts. We've done this for the progenitors in a previous paper, but this has not been done in great detail with the more differentiated cells. And so we're trying to understand the role of these proteins in adhesion and polarity, as well as interaction with various signaling pathways like the YAP-TAZ pathway. We're interested in deciphering the heterogeneity among ameloblasts, the mechanisms that drive transitions between secretory and maturation stages, and to identify novel regulators of mineral deposition. Want to understand the role of the various proteins during deposition at a single cell resolution. As Murali mentioned, we're collaborating with Tom's lab, as well as also working with the Payne and Hu labs to exchange reagents. And then lastly, I didn't have a chance to talk about this for the sake of time, but in parallel to the mouse work, we're spending a lot of effort, Dirk's lab is spending a lot of effort to build this biophysical atlas of enamel, which will also be another important tool that will be available to the community. And so I mentioned the folks who did the work as I was going through this. We also had help from several other members of the lab with various parts of the project. And we're beginning to work with Stefan Habelitz from UCSF on the amelogenin mutants. And thank you for your attention.
Thank you, Ophir. Our next speaker, Dr. Michael Payne. Dr. Michael Payne.
Right, thank you. Let's see. How's that? Is that up?
Yeah.
Okay, well, thanks everyone. Thanks Jason and Michelle for pulling this seminar together. I can probably skip through my slides pretty quickly in the interest of time, because a lot of what has been talked about by the other groups essentially has told you what the whole idea of this UG3, UH3 process is. So I do want to acknowledge the other groups led by Jan Hu and Jim Simmer and Dirk and Tom. And I particularly want to thank Tian Liang from the Simmer Hu lab, who has shared a lot of protocols and reagents. In my lab, I work with Joe Hussier and Yan Zhao, who are both faculty here, and Rucha, who's a recent postdoc and has done pretty much all of the work I'll present today, and Yan Bin Ji, who has been doing a lot of work with the animals. As Ophir mentioned, and Jason mentioned that this whole idea came out of a RFA many years ago. It was part of a workshop where people wanted to sort of increase the numbers of animal or models that could be used to look at enamel formation. And as Ophir has pointed out, for enamel groups, the Cree model of choice has been the keratin 14, and it has its disadvantages because it is expressed in a whole host of tissues. So as animals age, it makes it more and more difficult to sort of use these models. So for my group, we proposed in the UG3 phase 2 animal models, one being the ameloblastin Cree and one being the Odam Cree, specifically to look at secretory stage enamel events and also maturation stage events. We chose to use the PA2 IRES cassette that Ophir talked about. This allows us to express ameloblastin and enamel and Odam without any interruptions the way we planned it. And phase 2, we're planning to use, and I'll show you some data, both the ameloblastin IRS Cree cassette and the Odam IRS Cree cassette to look at a number of flux lines that have been used previously, primarily with the keratin 14 Cree, just to make sure that these models will be of value to the community. So the way we designed this, and I worked with the Jackson Laboratory to do this, so this was not done in the house, this was done at the Jackson Laboratory. So for the ameloblastin, the IRES Cree cassette was inserted immediately after the stop codon of ameloblastin, which ensures that ameloblastin is not disrupted as far as expression. And the idea with the Odam was exactly the same. The way the Cree was inserted was directly after the stop codon for Odam. So for the ameloblastin Cree animals, we then crossed them with the R26R cassette, and this is day 8. So let's see, this is ameloblastin at day 8. So at day 8, we see very little expression of the ameloblastin. There is a bit of background. I'll point out that we see this in all of our slides. Even though the teeth are clean at this stage, there is some background in the surrounding tissue. So, wait on. I made a mistake. This is actually the Odam Cree at day 8. So forgive me. Oh, sorry. No, this is ameloblastin R26R negative mice at day 8. So this is sort of a negative control, but we do see some background at this stage. So for the ameloblastin Cree at day 5, we see very little expression in the pre-secretory cells, but at the stage where we're getting to the early secretory, the laxate expression is fairly obvious, and it's at day 5, it's obvious in the ameloblasts of the first molar, but not the second molar. We do also see the occasional expression of ameloblastin in the odontoblasts, and this has been documented before, and then in the occasional pulpal cells. So this is ameloblastin at day 5, ameloblastin at day 8. It's a very similar picture where we see expression starting in the sort of the transit, in the sort of the early secretory cells, and then extending throughout the enamel. And you know, we missed the first molar in this slide, but you see the first molar in the top here, and you see expression. For the Odam at day 6, as the literature would suggest, we really don't see any expression of Odam at day 6. Whereas at day 11, we can clearly see expression in the first and second molars, some of the surrounding epithelial cells of the enamel organ. And here expression is starting, and this, if we had a higher magnification, this is sort of the transition stage from secretory to maturation. So these are the two models that we developed for the UG3 phase of the project. For the second part of the project, we've proposed to cross the ameloblastin Cree with three different lines, although two of them, SMUD4 and MAP kinase 14, has been published. So these two models have used the K14 Cree to study these, and they have an enamel phenotype. Jan, who didn't discuss it today, but she has LAMA3, and I think at this stage, because Ophir's ameloblastin Cree animal was available prior to ours, they've started working on this together. But these are models that we proposed with the ameloblastin Cree. The 4 models that we've proposed with the with ODAM Cree are STIM1 and STIM2, and RAC1 and CX43. And again, these are models that have used the K14 Cree and have an enamel phenotype. And we just, at this stage, really wanted to look to confirm that our animal models are doing this. I'm just going to briefly talk about the STIM1. This is an animal model now that we've, the Phlox STIM1 that we have data for. All the other projects are ongoing. Some we're doing in-house, and some of these breedings are being done by the Jackson Laboratory. So STIM1 is really the only animal I have data for now, but hopefully in the next few months we'll have some data for the others. So STIM1 we chose to work with initially. Rodrigo, who's not in the audience, I don't think, did a transcriptome profiling of both secretory and maturation cells, and we saw a significant increase in both RNA and protein during maturation. So we thought using the ODAM Cree to knock out STIM1 would give us a phenotype. It's a fairly severe phenotype when the K14 Cree is used, but as you'll see, it's significant, but not as severe using the ODAM Cree. So there are 3 papers that have looked at STIM1, one from Rodrigo, one from a Japanese group, and one from Petros Paparakis. And although with Petros, they've used the improved amelogenin Cree, which Ophir briefly mentioned in his talk. So we've got data for both animals at 8 weeks and 12 weeks. So at 8 weeks, the floxed animal, so it's the Cree, 1 copy of Cree and the double flox. If we look at the wild type, this is typically what we see, enamel that's very sharp at the end, whereas in the STIM1 knockout, the enamel is blunted. The other thing you noticed is that the knockout animal doesn't have the iron pigmentation on the surface of the enamel that you see in wild type. And I think the other feature that is fairly obvious is that in the floxed animals, the molars are more pigmented than in the wild type. And we think perhaps this is a post-eruptive sort of staining of a more porous enamel. So these images are pretty familiar to most people. We looked at the enamel density at 3 different regions of the teeth. And this was the data that came out. So if we're looking just distal to the first molar, with the mutant animal, there's a significant difference in enamel density. Just anterior to the enamel, it's the same sort of story. And by the time the tooth erupts, this is at the point of eruption, there's still a significant difference in the enamel. And as I say, this is at 8 weeks of age. And we did have samples at 12 weeks of age, and we did exactly the same analysis. And what we noticed here was distal to the first molar and mesial to the first molar, there was still a significant difference. But at the point of eruption, this significance we couldn't see. This was a sample size of 3 mutants and 3 wild types, and that's the data that we have for these animals. So I've mentioned the findings from these STIM1 mutant animals. There's a delay in enamel mineralization. The surface pigmentation on the sides is absent. The molars appear more pigmented or stained, and there's a blunting of the enamel tips. We're doing additional work. We're doing some gene expression profiling of these 8-week-old mice, and we're doing some SEM at the moment, and other sort of examinations are being proposed. And that is it. You know, as I say, in the interest of time, I'd like the discussion to go on, but this was the work that we've done. Both the animal models have been deposited in the MMRRC, and I understand they're sort of available at the moment. They've been breeding them at the MMRRC, and as soon as they're animals that they don't need, they're willing to share them. But you know, it seems that it's still been a bit difficult for some investigators to get these. So the animals are there. They can be purchased at the moment. It's not an immediate purchase at the moment. So again, I'd like to thank Jason, the DCR for funding, and thank you, everyone.
Thank you very much, Dr. Now, I give the floor to Jason for the Q&A and panel discussion session.
Right, so thank you all to all the speakers. As we open up the discussion, we do have a brief presentation from Rob Schuler, Dr. Schuler from FaceSpace. If you could share your screen and present. He's going to talk a little bit about the FaceSpace and how it's integrating into the data that these 4 groups that you've just heard are generating.
Okay, thank you, Jason. So we're very happy to have amylogenesis at FaceSpace. If you're not familiar with FaceSpace, we were established in 2009 with the mission to promote multidisciplinary collaboration and research in craniofacial development, molecular genetics, and genomics. We have developed and operate a platform for the curation and sharing of data that exemplify, quote, fair data principles. And if you're not familiar with those are a set of best practices and principles that are promoted by the informatics research community, increasingly have traction among journals and our funding agencies. And the FAIR stands for findable, accessible, interoperable, and reusable. So we are very glad to partner with the Amylogenesis Consortium and provide our infrastructure for the dissemination of their research data. So just to quantify the data that have been released or contributed and pending release through FaceSpace, of course, you have 4 projects in the consortium. We currently have 4 data sets that have been released and another 4 that are in preparation. By and large, mostly imaging data, although there are some other data and a little bit of sequencing data, about 684 files, images and related image-related data. Those also include a lot of detailed metadata, so descriptions of the experiments and the biological samples go along with that. On the right-hand side, what you see, I know it's a little bit small, so I'll kind of call out and describe it, but what you see on the right-hand side is a kind of a thumbnail of one of those data sets. So this is a data set that has been released to the public from this consortium. I would begin by pointing out that they are citable resources. So they come with full attribution of the project, the individual contributors. They are all registered with a digital object identifier registrar. So they have the DOIs that you would be familiar with from publications. On the upper right-hand side of those pages, there is a link there that shows anyone that comes to the site how to properly make a citation to that data set in their paper. Both gives them an example as well as a download that they can include in their reference. So we really want to emphasize that these are first-class scientific works. These are part of our overall scholarly ecosystem of communication. I also point out here that there are protocols associated with these. So in this case, I know it's small, but there are several, and these cover all the aspects, or can potentially cover all the aspects of specimen preparation, data acquisition, data preparation, and analysis that led to the data that you find on the site. And then you're really just seeing the tip of the iceberg here. There's a lot more detail on the experiments and biosamples. The key terms get rolled up into the next section that's labeled as standard terms, because these come from community standardized terminology sets called ontologies. So there are things like terms on anatomy from Uberon that pull together mouse anatomy, mammalian anatomy, zebrafish anatomy terms. The genes are labeled with the NCBI gene nomenclature with crosswalks to other gene nomenclatures as well, and so forth for the developmental stages, for phenotype. We have syndromes and a variety of other terminology sets. And as long as there are standard terminology sets available in the community, we adopt those in FaceSpace. And then next up are various forms of online visualization. So here you see in this data set, gross anatomy thumbnails. We also have a viewer for high resolution histology. So you can actually online go to some of these images if they've been shared through FaceSpace, and you can pan and scan and zoom around in those images on the site. We have a data processing pipeline in the background that transforms every data set and optimizes for online visualization. Of course, the original data is always preserved, and that's what you download. So lastly, I would just say that data that comes in through FaceSpace, again, is curated by this group, by the members of this consortium, according to a data model that really makes possible this fairness of the data. So all of the data are documented with the project, of course, the group that it came from, the data sets and that high level description. Then within that, the details on the experiment, so the types of assays, linkages to the specific protocols and the documentation that comes along with that, the specific instruments that were used. There's a variety of different imaging modalities, for example. And then details on the biological samples and the replicate groupings that are involved in each of the experiments, so the species and genotype and so on details. And then every single file that you find will be linked into that, so you know exactly what it's a part of. And these FAIR principles, as I mentioned, they're increasingly important to scientific journals and our funding agencies, and they really make possible the reuse and reanalysis of this data, so that you can be assured that data that's produced by this consortium will come with rich descriptions for your usage. And that concludes my presentation. Thank you, and we're appreciate being part of the consortium partners here.
Thank you very much, Rob. We're very happy to have FaceSpace involved in storing and making this data available as a knowledge base to the community. Now we're, I guess, opening this up to questions from the audience.
I had a quick question. Ophir, you mentioned that you had some mice in the depository, and could you name the depository and the mice? For example, do you have an amylogenin Cree mouse that's been tested
So they are registered at MMRC, and I'm not sure if they are. I'm not sure which ones are ready yet to go. So our plan is to put everything that we have in there. Right now, if anybody wants anything, we've already sent some out, and we're happy to just send them to you. It'll be quicker and cheaper than getting them from MMRC. I think that they will be. I don't want to actually say the exact date because I have to double check. So we have there's a number. Like if you look on their website, you'll see a number registered for us, but that doesn't mean that necessarily that they're all expanded and ready to go yet.
Well, that's generous to send them directly to us if we want them. I appreciate that. Have you tested them?
Yeah, I mean, it's a very small community. It's not like we have like thousands of requests for these things. It's like, a
Yeah.
few people asking. It's easy to send them.
Well, I think in one of your first slides, you showed that they cut out beta actin or something like that, or
No,
was
the
that?
beta actin Cree was to recombine the flox alleles.
I see.
The Crees that we've tested with reporters, and they all seem to work as the ones that are completed all seem to work as expected. We did have one locus that was challenging and that we had to retarget a couple of times. But the ones that we have right now, like, if the amyloblastin or KLK4 Crees, those are ready to go.
Okay, great. Thank you.
Yeah. Yeah, we're planning. We have the initial draft of figures together that we want to send out as sort of a resource to JBR or another journal, and once I have that ready, I'm also happy just to send you that if you want to look at the images from that.
Thank you. Appreciate that.
Yeah.
Thanks for your question. Just to reiterate, yeah, so the intent of these, this consortium or the groups of the from the UG3 and UH3 were to generate these models and to have them freely available to the public, or provided them through the MMRRC. So the link is in there. And as you've heard from the investigators, they are also available from the PIs if you contact them as well. And of course, for the data part, FaceSpace is always open. And I see a hand from Stefan.
Hi, everybody. I just had a question about one of these mice from Jan Hu. That's the modified citric transporter, or the knockout of that LFC transporter. And I was wondering about the mechanism. Is there any relevance to the pH homeostasis, control of pH, since citrate is involved? What's
At this time, we don't know yet. The primary function of citrate is largely deduced from other system, and within the aminogenesis process, whether there's influx or efflux of citrate into the enamel matrix, we still don't know. And so that makes the model attractive. If your laboratory is interested in investigate that area, we'll be happy to send the model.
Sounds good. Thank you.
You're welcome.
So speaking of collaborations, some of them mentioned collaborations within the cooperative agreement groups, and I'm sure any of them are very happy to collaborate with those in the community.
Yeah, I mean, I'll just say in our case, we've made so many mice, like even if we wanted to, there's no way that we're going to be able to use all of these. So we're really hoping that they get adopted by people and find good homes at their labs.
Michelle has pasted in the chat the links to the MMRRC for the mouse models, and as well as to facespace.org where the data is being housed. So please check those out.
Yeah, and it would be I'll give my experience with the MMRSA. Our mice were deposited probably about 12 months ago, and because they sort of go through the process of freezing down embryos, they keep some stock alive while they're breeding them, and if they're retiring these animals or they don't need them, they'll make them available. But I know that Pam Dembeston tried to get the Odam Cree from the MMRSC starting probably about November last year, and they said they'd be available by February. I think she got a delivery last week, so it did take a bit of time, but hopefully at least for the two animals we've got. These will be fairly freely available from here on in. I think they've probably got their stocks now. And because we didn't make them in-house, we actually bought the Cray animals that were crossed with the R26R. So we had some of those, but we only had them with the R26R, and now we've sort of got stocks for the Odam Cree in-house. We're doing all the floxed animals with Odam Cree in-house, and with the Amyloblastin Cree, is work that we've contracted out to the Jackson laboratories. So we've sort of split up the work between the two groups.
I see Joy put in the chat
Yeah.
something about
Yes.
yeah.
Yeah, I don't know if you're already considering this, but it would be great to sort of put it all together, especially some of the initial characterizations that you've already done that, you know, help the user to really be able to move on to the functional studies quite quickly. Like you've already proven that, you know, the expression is exactly where you expect it to be.
No, so that's
that,
exactly what we're planning to do. I think because we're looking at this as sort of a suite of tools, and we tried to be consistent in how they were generated, and you know, at least in the ones that we made, we're trying to sort of group them in categories and explain you know why we made certain decisions in terms of generating them. Like I was talking about, you know, the knock-in, knock-out versus P2A and things like that. And we also tried to be thoughtful, though it was tricky, with choosing reporter colors so that people could cross things in various ways and get you know, multicolor. We call it like enamel bow or something like that, like the rainbow mice, so that you could have, you know, the different stages in you know, the correct colors. Yeah, our only holdup right now is that there are about 10% of the lines have been tricky. We actually had to, one of the loci that we chose didn't work, so we had to choose other loci and target there instead. And so we might, in our case, we might just go ahead and say, well, if we have almost all of these reagents ready, we might publish those and then publish the other ones in the future, just to not hold it up. But we're hoping that in the next, you know, period of weeks that we'll know a little bit more.
That sounds great. Yeah, wonderful symposium. Thank you, guys.
Thank you. Cool. Well, thanks, Michelle and Jason, for organizing this.
Oh, you're welcome. Yeah, thank you all to the speakers. I guess we don't have any more comments. Any last minute comments or questions from the audience? No? Well, I guess on behalf of Olivier has a comment. Great to see all these new tools. Thank you. Thank you very much. I guess on behalf of Michelle and Cortez and I, as co-chairs of this session, and of course to the mineralized tissue group for their sponsorship, and as well as to you know, support of NIDCR for these groups, we thank you for joining this session. And please feel free to reach out to any of these PIs. They're more than happy to share these models and resources they have created, and as well as the data. There's going to be a wealth of data. There will be a wealth of data that will be generated. That's all freely available and accessible to all of you. So please avail yourselves of all that data. I'm sure there'll be lots more research avenues to come from that. So thank you very much and have a great day. Michelle, do you want to have any?
Thank you for everybody for attending and for the speakers for your amazing presentations, and let's hope to see more things coming out. So that's it. Have a great afternoon.
Thanks, everybody.
Thank
Thank
you.
you, everyone.
Bye-bye.
Thank you,
Thank you.
everyone.
Take care.
Bye-bye.
Thanks. So I guess the recording will.
Stop, right? You?
Okay, yes. So we can stop and leave.
Yeah. Okay.
Okay.
That
bye.
was great. Bye bye. See ya.